Okay, welcome everybody. So we will start, it is a bit late, but we will start the next session on chromosome mechanics. We will have 3 wonderful talks on 3 different topics related with the chromosome dynamics and so on. And the first speaker will be Ana Losada. Ana will be talking about cohesion and cohesin. This is her specialty for many years, and she is the principal investigator of a group that has been for more than 20 years working on the CNIO.
For more than 20 years at CNIO?
For more than 10 years.
Ah, I'm not that old. I am old, but. Okay, so while the slide comes up, I just want to thank first Alberto for inviting me to present my work here. I'm sorry I couldn't be here yesterday, but I will be here until tomorrow. So I expect to see also the work of colleagues. So the work I'm going to tell you about is not on cohesion, but it's on cohesin, and it's about the role of cohesin variants in 3D genome organization. And it's the work of a staff investigator in the lab, Ana Cuadrado, and 2 PhD students, Alec Kojic and Magali de Koning. So the way we understand the organization of the genome has been changing recently. So we knew from a long time that the genome is organized in a hierarchical way, but the recent development of computational methods related to chromosome conformation capture and high-resolution microscopy has challenged the vision that we have of the genome. And one of the major organizational principles of the genome has come to be these chromatin loops that put in contact 2 distant sequences in the linear sequence. And these loops can be of many types, involve polycomb proteins, or bring together enhancers and promoters, and so on. And then there is a superior organization, which is the chromosome domains, or TADs, topological associated domains, in which you have loops within loops. And then you have the compartments, and then you have the chromosome territories. And all this organization is important for proper gene expression in the cell. And so I want you to notice this circle here, and this is cohesin. So cohesin is a protein complex that I identified when I was a postdoc in Cold Harbor many years ago. And it is a member of the chromosome SMC, the family of chromosomal TPases, SMC, for structural maintenance of chromosomes. SMC3, SMC1, there is a glycine subunit called RAD21, and then SA. And there are 2 additional regulatory subunits that we call PDS5 and WAPL that are going to modulate the interaction of cohesin with chromatin. And cohesin was initially identified for its role in sister chromatid cohesion because this complex has the ability to bring together 2 pieces of DNA that can be the sister chromatids. And cohesion is essential for proper chromosome segregation and also to mediate DNA repair by homologous recombination. But in recent years, it has become apparent that cohesin can also entrap the 2 DNA pieces that are in the basis of a chromatin loop, and therefore it promotes proper genome organization together with an architectural protein called CTCF that I will mention later on in my talk, and in this way also regulate the interaction between enhancers and promoters and regulate gene clusters and so on to mediate proper gene expression. So how cohesin works, it's still not very clear. But one thing that we realized when we started looking at the distribution of cohesin in the genome is that if you look at the position of NIBL, which is the protein that loads cohesin onto DNA, there is no cohesin. This is just a mapping of the chip rigs along the NIBL summits. There is no cohesin here. However, there is a huge colocalization between SMC1, cohesin protein, and this CTCF that I just mentioned. So it is believed that once cohesin is loaded onto DNA, it can move along this DNA to reach other positions until it reaches these CTCF sites. And there is a model put forward recently by a number of labs using a lot of experimental data, but also computationally and mathematical modeling that suggests that cohesin can, once loaded onto DNA, extrude DNA, it's unclear how, and form these chromatin loops that are eventually stabilized at the sites in which two CTCF binding sites are meeting each other. And in this way, it stabilizes these loops that can be later seen in this high-C analysis in which we compare the contact probability between two regions of the genome. And so the thing is that there are not all the cells in your body have cohesin, but there are variant complexes, variant cohesin complexes. And the variant comes from two sides. One is that the SA subunits can be SA1 or SA2, and the PDS5 regulatory subunit can also be PDS5A and PDS5B. And these are, in both cases, these are hit repeat proteins, and they are similar, very similar to one another. But one cohesin complex can only contain one of each. And so we were wondering about what is the functional specificity of these different cohesin complexes that are present in our cells. And in order to explore this, we decided to generate some mouse models for deficient for some of these proteins. So we generated first PDS5A and B, and I won't talk about this today. And a number of years ago, we also generated a knockout for SA1, and more recently, in collaboration with Paco Real at the CNIO, we have generated a conditional knockout for SA2. And I will briefly mention some of the results that we have. So in terms of development, what we noticed is that the SA1 knockout mouse the embryos started to die around 10.5, 11, but we could definitely get MEVs, and we could even get some late embryos that were clearly smaller and had like a general delay in development. When we look at the MEVs, we were surprised to see that there were no cohesion defects in general in the chromosome. However, when we look at the telomeres, we did find defects in telomere arrangement. And this is what is called telomere fragility and comes from defective telomere replication. So this is published, I won't bother you with that, but just to say that cohesion at telomeres is important for proper chromosome replication of these ends, and in the absence of this proper replication, you have chromosome segregation defects and some slow proliferation. Now, in the case of the SA2 knockout mouse, the embryonic lethality starts to be a bit earlier, around 9.5, 10.5. We are trying to exactly point out what is the reason for this lethality, but you can see here 10.5 wild-type embryos and knockout embryos, which are clearly, I think they are almost dead, at least this one. And so we cannot get MEVs to study cohesion in these cells. So we also have, we combine the conditional knockout with a CRE recombinase that is inducible by addition of tamoxifen to the culture media. In this case, after 4 days, we can eliminate the SA2 protein. And you can see here that elimination of SA2 brings up a little bit cohesin SA1, to compensate, so the final amount of cohesin is not different in the wild-type and the SA2 knockout cells. So when we characterize these cells, we found that there is some slower proliferation, but it was much minor defect that we expected. And also, we found that there were chromosomes in which the cohesion between the centromeres was lost. And this was, again, more subtle than we expect. So more of the defects that we observe are what we call mild defects, in which there is not a complete separation of the chromatids, but the chromosomes are still together, but the centromeres are a bit split. And as a consequence of these defects, there is some problems in chromosome segregation in the form of these anaphase breaches or lagging chromatids. But again, this is not as huge as we expected, so it seems that there is a huge compensation between cohesin SA1 and cohesin SA2, and there are no problems in proliferation and in the cell cycle that we can observe. So the important thing here is that cohesin mutations have been identified in cancer over the last 5 years. And one of the genes of the cohesin genes most frequently mutated is the one encoding STAC2, which is the SA2 subunit. And this is particularly important in bladder cancer. This is work from Paco Real at CNIO. but also in myeloid cancers and even sarcoma, there were many cohesive mutations. And the reason why we think that STAC2 is most frequently mutated is double. One is that STAC2 is on the X chromosome, so a single hit is sufficient to eliminate the protein. Most of the mutations that are found are loss of function mutations. And then we think that in the absence of SA2, SA1 has the ability to perform cohesion, which is essential, and so the cells can survive. However, we speculate that cohesing SA2 may have some specific function that might be altering these cancer cells and is providing some kind of advantage for the tumorigenesis. So we wanted to understand a little bit more what is the specific function of cohesing SA2 in gene regulation and in genome organization. And in order to do that, we started by, we selected a primary human cell line in which the amount of cohesing SA1 and cohesing SA2 were similar. And we did ChIP-seq with 3 antibodies, SA1, SA2, and a common subunit, SMC1, and then we obtained data, public available data for CTCF. And this is just to show you that we found like 3 classes of positions. In up to 40,000 positions, we find that cohesing SA1 and cohesing SA2 coexist. Of course, SMC1 is also there. And these positions all contain CTCF. But there are also a large number of cohesing positions in which there is barely SA1, so this is why we call them cohesing SA2 only positions. And in most of these positions, there is no CTCF. The SA1 only positions are very, there is a small amount, but they are very similar to the common. So what I mean is that although they don't have SA2, very little SA2, they have some CTCF. And so when we check where in the genome are these complexes, we find that the common positions are mostly distributed in what is called insulators. So insulators are regions characterized by the presence of CTCF. So this is logical because there is a huge overlap with CTCF. And are also present in enhancers, which are here in this yellow, and a part of them are in promoters. In contrast, the SA2 only positions are mostly present in enhancers and promoters, where, of course, consistent with the fact that they are not colocalizing with CTCF, they are not present in insulators. So this was a striking difference. SA1 only positions resemble more or less the common positions. And when we did the analysis to check what are the binding sites, whether they contain some transcription factor motifs, in the case of the common positions, again, the major motif is CTCF. It's not that cohesing is binding the CTCF motif, it's that CTCF is there at the same time as cohesing is there. And only in the case of the SA2 only positions, we start to see motifs for other transcription factors. And doing some proteomic analysis that I won't have time to show you, we also find that SA2 is more versatile in the partners with which it interacts, and it interacts with a number of transcription factors. So this is not specific of these mammary epithelial cells. When we do the same type of ChIP-seq analysis in the cardiac endothelial cells, we observe the same kind of distribution, common sites, SA2 only sites, and in this case there's a larger amount of SA1 only, but with similar characteristics. And when we compare these two distributions, we observe that the common positions are similar in the mammary epithelia and the cardiac cells. However, the SA2 only positions are more different, suggesting that SA2 only positions have a more important, are driving somehow cell-specific programs. And so there's one particular kind of enhancers that it's very important to promote proper cell identity, which are the so-called super enhancers. And these enhancers are close to the cell identity genes. So they are present they are driving for example, a cell identity gene. And here you have another, whereas in the typical enhancers, they are closer to the genes and not in one or the other. So when we look at cohesin distribution in these super enhancers, which are defined in each cell line, we can see that all super enhancers contain cohesin, and that there are common positions, but there are also a huge amount of SA2-only positions. And just let me show you an image of the browser to show, so this is a particular super enhancer that is working in the mammary epithelial cells. So on both sides of the enhancer, we find the cohesion common positions. So we have SA1, SA2, and SMC1. But in the middle of the enhancer, there is very little SA1. There is a lot of SA2 and SMC1. But this is particular for the HMEC. When this super enhancer is not working as such in a different cell type in the CAIC, these positions disappear. So there's an important correlation between the presence of SA2 and the activity of the super enhancer. So we wanted to do some functional analysis depleting the proteins to see what happens in these cells, and the primary cells are not good for transfection. So we turned to these MCF10A cells, which are also mammary epithelial, but are immortal, although non-tumorigenic. And first we checked that all the characteristics that we have found in the primary cell line were conserved here, and indeed, again, you have the common sites that have CTCF, the SA2-only sites don't have SA1, and only a fraction of them have CTCF, so probably this could be common sites that are mislabeled here. And again, the common sites, insulators, enhancers, where the SA2-only are mostly enhancers and promoters. So everything was consistent with our previous analysis. And so we use old technology, siRNA, to downregulate SA1 or SA2. And we bring down cohesion to similar extent. As control, we also downregulated SMC1, a common subunit. And then ask what happens with gene expression. When we did RNA-seq and compare what happens, we also, for control, we also compare with what happens with CTCF. When we did RNA-seq to analyze how gene expression changes in these cells, so we observed that most, the cells that were changing more the gene expression with were CTCF downregulated cells. But then the SA2-only cells were also changing a lot. And importantly, there was only a partial overlap between the two gene sets. The SA1 have also overlaps with CTCF and with SA2, but the changes were much minor. And so when we look a little bit at what changes were going on in here, so we observe the ones that caught our interest is in the case of the downregulation of SA2, we found that there were genes that shouldn't be expressed in these cells because these are mammary epithelial cells that started to be expressed. And in particular, also genes of the hematopoietic lineage. And this is important because, as I told you, among the cancers that are frequent having STAC2 mutations are the myeloid cancers. And this set of genes were not found regulated in the same way when we change it, when we downregulated SA1 or when we downregulated CTCF, suggesting that SA2 had a function that was independent of CTCF in the regulation of gene expression. So we decided to look at if we could correlate the changes in gene expression with the changes in distribution. And we first selected a locus in which we had found a number of genes that had SA2-only positions at their promoters, and they were downregulated when the genes were downregulated when we eliminated SA2. And this is this gene cluster, which is the S100A gene cluster, that contained many genes, and that we like it because it had the two kinds of positions. So they had strong positions that were common. They contain SMC1, SA1, and SA2, and these are, we have signaled some of them. I don't know if you can see them with C for common positions. And then there were other positions that were smaller, for example, this one, and there were only SA2-only positions that contain SMC1 and SA2, but not SA1. And this happened in several parts of this cluster. And we first checked by ChIP-qPCR that what we saw in the ChIP-seq analysis was true. And again, so the common positions have SA1, SA2, and all of them have CTCF here in gray, where the SA2-only positions are usually smaller, so they are probably less frequent in the cell population, more variable. They contain SA2, but not CTCF, not SA1. So The important thing is that when we downregulate cohesin SA2, what happens is that in the common positions, cohesin is maintained. So this is a fold change, so one is no change between the cells treated with control siRNA and with SA2 RNA. So in the common positions, there were no major change for SMC1, whereas in the SA2 only positions is when we started to see a decrease as we expect. I must say that in this case, we had some artifact because, for example, here when we downregulate SA2 and we chip SA2 in the SA2 cells, the signal not only doesn't decrease, but increases. And this is because the antibody now has less sites to go to, so the sites that are remaining there are easier to chip, so the signal is increased, okay? And the other important thing is that when we downregulate cohesin in general, we again see the same, that SA2 only positions decrease, so these are easily removed, where the common positions are more resilient. So of course this is an siRNA and it's not 100% elimination, so we always have some cohesin present in the cell, and what we think is that this cohesin that is remaining is the one that is located in these strong and common positions. So from this we also conclude that since when we downregulate SA2, the signal of SMC1 diminishes here. This means that SA1 cannot go there and compensate the lack of SA2, okay? So this is the first conclusion from this data. Cohesin SA1 is unable to go to the cohesin SA2 only positions. Then we make one prediction, and this is that the loss of SA2 is likely irrelevant for the function that cohesin exerts from these common positions, since we see that this cohesin is more resistant to depletion. And then we have one question: what are the difference between this common and SA2 only positions? Can we characterize them in any way? And in order to test the prediction, we decided to do a 4C analysis in which, so we had data from the HMEC cells in which we see that in this locus there are like two subtats separated by this region. So we set our viewpoint in this region and we check what are the connections that we see with other regions of the genome. And you can see here that when we set here the viewpoint, most of the contacts are within this subtat. When we eliminate SA2, this profile doesn't change, it's virtually the same. However, if we eliminate CTCF, which is the architectural protein that is essential to set the boundary, then we start to see that this guy is seeing sequences present in the neighboring TAT. So I think this makes our case that, you know, the cohesin SA1 that is present still when we eliminate SA2 is sufficient to promote this boundary between the subtats. So then when we started to characterize what is the difference between the SA2 only positions and the common positions, and sorry because it's very contrast, so I'm not sure that you can see, but if we plot the reads around the summits of the peaks, I hope you can see that the SA1 only position or the common positions make very sharp peaks. However, the SA2 only, which are here in blue, are peaks that are broader. And for us, in a very naive way, this suggests that SA2 can move a little bit more around these positions. The other thing is that I told you at the very beginning that there are two regulatory subunits, PDS5 and WAPL, that interact with cohesin and regulate a little bit the dynamics of cohesin. And so we decided to look whether the amount of WAPL, which is important for this dynamic behavior, is the same in the common and the SA2 only positions. And we found that that is not, that there is a difference. So while in comparison, the relative amounts of cohesin and WAPL in these common positions is much different than here, suggesting that this cohesin is more frequently bound to WAPL, and it's therefore making cohesin more dynamic. We also found that in our proteomics analysis, when we IP from two different human cell lines with SA1 and SA2 antibodies, the amount of WAPL that we see in the SA2 is always larger, and it's larger, so there's always a better IP of cohesin SA2 than SA1, but the amount of WAPL that we see is even larger than that, again suggesting a preferential interaction of cohesin SA2 with WAPL. And then a low-tech experiment, which is a salt extraction experiment. You obtain the chromatin from the cells, you treat them with salt and check what proteins are being removed by increasing the amount of salt. And I hope you can see here that when we treat the chromatin of wild-type cells with a bit of 0.5 molar salt, cohesin, a common subunit is reduced, of course, but the reduction affects much more SA2 cohesin than SA1 cohesin. And when we do similar analysis, in this case in the MEVs that are knockout for SA1 or SA2, when we have SA1 knockout, so the cohesin that we have here is SA2, there is an stabilization because now this cohesin is assuming the role of the one that is gone. And the other way around, when we eliminate SA2, so the only cohesin is SA1, the cohesin is more stable. So the general message here is that cohesin SA2 goes more with WAPL, is more dynamic, moves around more, it's easily extracted, whereas cohesin SA1 tends to be closer to the CTCF sites, and we think that this stabilizes the protein, makes it more difficult to be extracted, and it's made these sharp peaks. So we have noticed before, when we characterized the SA1 knockout MEVs, that, so in the MEVs we also find if we chip SA1 and SA2, most of the positions are common, but we also have SA1 only, SA2 only. And the most interesting thing that we found a few years back is that when we IP cohesin in wild-type and SA1 knockout MEVs, the number of positions that we can detect in the SA1 knockout MEVs increases, and increases quite a lot. And these new positions don't have CTCF. And again, this is consistent with the idea that cohesin has a tendency to be here with CTCF. If you remove cohesin, the cohesin that is left, which is SA2, although it can still go to common positions, it is more mobile and it's more independent from CTCF. And now when we chip cohesin and we compare the wild type with the SA2 knockout MEVs, we found exactly the opposite. So now the number of positions that we find in these MEVs which only have SA1 is reduced to the wild type. So I think all this points to the idea that not only that cohesin SA1 prefers to bind CTCF, but somehow cohesin SA1 binding to CTCF attracts cohesin SA2 to these common positions. So we wanted to explore this idea a little bit more. And what we did is to check what happens with cohesin SA2 at common positions when we eliminate cohesin SA1. These are two negative regions. There are 5 different common sites. This is the amount of cohesin that we find in the this is SA2, sorry, that we find in the wild types, and it's very much reduced when we eliminate SA1. So we speculate that cohesin SA1 comes first. It arrives to these CTCF sites, and then the next cohesin that is loaded is somehow trapped by this cohesin SA1 that is there. So in order to prove this model, we want to demonstrate that we can find more than 2 rings in a single position. And of course, in a population, we cannot do that, but since we have SA1 and SA2, we decided to do redshift analysis. And we could indeed find that when we IP first SMC1, which are in 5 different of these common positions, this is the first chip with the first SA1, and now we get this chromatin and we chip with SA2, and we see that in all cases, there is a high background. I must say the third line is the background because we are chipping after the with IgG after the SA2, but there's always higher binding above the background. So, and the same in we IP first SA2 and then SA1, suggesting that this model can be true and that at least 2 different rings can be found in a single position in a single cell. So we speculate, taking this together with the previous slides, that cohesin stacking at this position may be important to promote their unloading and therefore stabilize the cohesin that is present at these sites, and therefore we get these common sites in which the number of reads that we get when we do chipsec is larger than in the other SA2 only positions. So this is a bit of a summary of what I just told you. So we think that cohesin SA1 and cohesin SA2 have somehow specialized roles in genome 3D organization, and SA2 is more involved in local chromatin contacts. So this relates to the figure at the beginning. So this would be like a TAT, and then you would have numerous chromatin loops within a TAT. And cohesing SA2 would be important for mediating these local intra-TAT loops. And this would be more transient, more sensitive to depletion, lower peaks. However, in the domain borders, you would have CTCF, of course, and you would have SA1 and SA2 in many cases, larger peaks, more resistant to depletion, less WAPL, more stacking, and this would be the ones that really make the organization in the contact domains. My final slide is, so we wanted to prove a little bit further whether this was the case, so we decided to do a high-C analysis in these depleted cells. This is my final slide. And so what we have found, and I won't bore you, so what you're looking here is a matrix of contact probability in which you plot here the same as here, so you are checking contacts along the diagonal, okay? And so in this particular, this is chromosome 15, and we are checking differential interaction between SA1 depleted cells and control, and here SA2 and control. And what we see here is that in the SA1 knockout cells, the shorter interactions, which are in the diagonal, are decreased, therefore they are in blue, and these and what increases is the more long-range interactions, we are here in red. And the opposite happens in the SA2. So when you deplete SA2, you increase the interactions in the short range and deplete the interactions in the long range. So we think that. I forgot to mention we did this with Mark Matirenome in the CRG. So we think that this kind of provides further proof for this model of cohesing SA2 being more important for these intra-TAT interactions, and cohesing SA1 being more important for these domain interactions. So let me just finish by acknowledging again the people who did the work. So Alec and Anna carry out the main part of the work with some help from Magalin, which is the one that developed the SA2 knockout MEFs together with Paco Real group. And as I said, Mark is helping us with the high-C analysis. And I will just leave you with another version of the model in which, again, you have several TATs, and you have SA1 and SA2 in these common positions, and SA2 is here. So when you deplete SA2, for example, you see increased number of intra-TAT interactions, but probably these are stochastic interactions, not mediated by cohesion, and these deregulate gene expression within these loops. So I think I will stop here and just take questions.
Thank you very much, Anna, for this beautiful talk. Then we have questions. Let's start.
Yeah, beautiful talk. So I was wondering, in your specific mutants of the subunit, did you try to do high-C to kind of quantify what is the global effect on TATs, if there is any?
You mean in the MEFs, in the knockout MEFs?
Yeah.
No, we haven't done that. MEFs are I don't like these cells.
Yeah, they are not nice, actually.
So that's why we turn out to the human cells. What we are doing now, we are trying to do this in the mouse CS cells, mutant for one or the other, to see. We expect to see similar results and better define the correlation with gene expression, which is also very difficult to assign. Yeah.
And
but it's the final goal.
Then another question. So probably you will have, I mean, in different cells you will have maybe different compositions of the subunits. So do you expect to see like differential effects depending on the cells where you are looking at?
So what we have found, so before choosing the mammary epithelial cell lines, we screen a large number of cell lines just to see whether, we wanted to do the analysis in a cell line in which the amounts of the two variants would be similar, so the quantity would not prevent to analyze the quality. And the truth is, it's in different cell lines, you have a slightly different ratios. In general, there is more SA2 than SA1. HMEX are the ones in which they are more similar. I don't remember which are the ones that are less similar. But I think the general scheme is going to be the same. Then there are cells that express many more genes than others. So So we had also one analysis that we did in comparing in the mouse, in the mouse pancreas and brain, for example. The pancreas is less complex than the brain in terms of gene expression, and this was reflected also in the number of positions that we have for cohesin. So there is a close correlation between gene expression and distribution of the complex, and it goes two ways. So depending on how much complexes do you have, yes, we expect to see a slightly different effects, but the overall, I think it will be conserved.
Then Anna, I'm here. I mean, you show beautifully that when you remove, I think was SI2, then you didn't have CTCF recruitment, therefore SAA1, the conclusion was that SAA1 was not replacing that. But I guess, I mean, it would be nice in any case to check a cheap distribution of, let's say, SA2 in first depleted of SAA1, because it might be that this may not be the same behavior in all of the genome, so that in some cases one might replace the other. And that together with, well, let's go.
No, for sure. I mean, I'm showing you the extremes, right? So
yeah.
I'm showing you the ones in which there is a clear replacement. But there has to be some conversation between the two complexes, because the embryos, they get to 10.5 in the case of SA2 knockout. So there is some conversation for sure.
And then it would be worth to do SAA1 and SAA2 depletion and check what is the impact on expression, because you have redundant action in some genes, but specific one in some others, but this may be changed. I don't know whether, I know that the genome-wide analysis, you can go over and over and over, no. many multiple combinations, but...
No, it's true. So instead of that, what we did is we did SMC1 depletion, which is you are removing the common subunits. So in principle, you are removing more or less both. The problem is, I don't like very much the gene expression analysis involved, because there are a lot of secondary consequences. Okay, so it's difficult to assign which are the true consequence of the removing one or the other. So what we're doing now is, as I was replying to the first question, we are doing now mouse CSLs because there, I think it's more clear that we are going to affect developmental genes, which at the end of the day, the field is, it's clear that these are the ones that are going to be more affected by these enhancer promoter contacts. And we expect to see this a more clear picture of what happens when you remove one or the other or both.
And just to finish, because this is the final message, no? That is the way that these cohesins are working in a super enhancer, is because you are kind of helping folding the DNA to do the action. But then I guess that the best way to get into that is to go into a specific gene and check that directly, no? Rather than genome-wide, to learn how that works.
That's the way we have to do that. And we want to do that in the context of polycom chromatin, yeah.
Any other questions? Then we can, yes, of course, Mr. President.
You can't escape to ask that. Maybe it's too early for this question, but I'm curious to know. How do you envision that the cohesions are physiologically removed from their binding positions? Because there must be a dynamics of the system, right?
So there's a lot of work going on right now to try to understand how cohesin loads, how cohesin unloads, and how cohesin moves. And this loop extrusion model that I put forward there, it's unclear. These guys are chromosomal ATPases. The ATPase is very important for whatever the complex does. The complex has two cavities. One is, so the way I usually draw cohesin, it's like a single ring, but the truth is that the heads of the SMC can be open or closed. So there is two cavities. There's one cavity here, and there is another cavity between the two closed heads and the glycine subunits. And it's unclear how this ATPase, which regulates the opening and close of the heads, is going to modulate the opening of the ring. But there's a lot of, there's beautiful work going on the structural level. So right now there is a boom of structure and single cell analysis, TERF, to see the cohesin move along DNA. So I think in the next years we're going to learn quite a lot about how these proteins work.
Then thank you very much, Anna. Then we move to the next speaker, Antonio Giraldez. Antonio started his career here in Spain, but then he moved from Europe to the States, and after being in different labs from Heidelberg to New York, then he got his lab in the University of Yale in New Haven, where he's the director of the Department of Genetics. He's one expert in gene regulation in embryonic cells, and then we are very glad that he's here to talk about his research.
So it's really a pleasure to be here. It's my first time in the north of Spain. I'm from Jerez de la Frontera, and it's actually interesting. I realize it's one of the few meetings that they pronounce my name correctly, so thank you. So, and it's really a pleasure to be here and really interact with former colleagues and really see this beautiful part of the country. So today I'm going to tell you a little bit about our interest in gene regulation and try to decipher the code by which the embryo really regulates gene expression in the first hours of development. And we're interested in understanding a universal transition that happens in all animals, which is the maternal to zygotic transition, by which upon fertilization the genome is typically transcriptionally silent, and development is controlled by thousands of mRNAs deposited by the mom into the egg. So in this silent genome, these maternal products are driving the first hours of development, and depending on the species, that varies significantly. And those genes are also responsible for the activation of the genome. And upon the activation of the genome, there is 30 years of knowledge that actually this results in the regulation, post-transcriptional regulation of those, many of those maternal instructions in a way that the embryo can transition from this maternal stage to these zygotic stages of development. And you have to imagine that this is really a profound developmental transition, where it's one of the most profound remodeling of gene expression, where you have thousands of mRNAs that need to be degraded and translational repressed, and thousands of genes that need to be activated to install the new program. And another reason why we're interested in this process is because it really represents a transition, and that is reminiscent of every transition that happens in development, where you typically have to install a new program and also remove the previous program. So we think that by understanding the basic principles by which the embryos undergo the activation of the genome and the post-transcriptional clearance of the previous messages, we can learn about basic processes of developmental biology and disease. So I work in zebrafish development mainly, and so this is a zebrafish embryo, early development, and up until here, all the processes that you see are developmentally controlled by the maternal instructions. And now here, the genome become activated, and you see the different movement of the cell. So you go from this rapid synchronous cell division to then an organization of the cell movement and activation of the zygotic development. And this is and this is about, I would say, a 6-hour movie. The second step is the clearance of the maternal products, and this is just to illustrate an in situ hybridization of one maternal mRNA that is clearly degraded after the zygotic genome becomes activated, and this is actually blocked if you block transcription of the genome. So some of the questions that we're trying to understand in the laboratory is, what are the mechanisms and the factors that mediate the activation of the genome? What are those maternal genes that are responsible to go from a silent to an active genome at the right time on the right genes? And second, what is the post-transcriptional regulatory code that mediates the clearance and repression of many of those maternal mRNAs? So, so to try to understand what are the factors, we hypothesize that, well, these maternal products need to be highly translated because you have a high activation of the genome of many different genes. So we actually performed ribosome footprinting and found that NANOC, OC4, and SOXB1 family transcription factors are the most highly translated sequence-specific transcription factors in the early embryo. So now we, in a published work that I will not go through, we perform loss of function for these factors, and observed that they are required for the global activation of the genome and to initiate zygotic development. So if you block the activity of these factors, you block the process of gastrulation and you block the process of the clearance of maternal mRNAs. And indeed, one of the factors that is activated by NANOG is this microRNA miR430 that I discovered during my postdoc that is responsible for the post-transcriptional regulation and decay of about 20% of the maternal mRNAs. However, we were never able to really get a grasp of the timing. What regulates the timing of the activation of the genome? Why the genome is activated at 2.75 hours, not before, not later. So, and this was a project that was tackled mainly by Henry Sucheng with the collaboration with several other researchers in the laboratory. And the idea is, can we understand what controls the timing of this event? And this has been a long-standing question, and there have been many different models proposed in the field. One is that because you have very rapid cell division, you only have S and M, and you don't have G phases. There is a clash between transcription and DNA replication, and that therefore maybe inhibit zygotic genome activation. There was a proposed model by Mark Kirchner where they, because the genome activation happens after several cell divisions, and these are reductive cell divisions that reduce the cytoplasm and increase the number of nuclei, proposed the model of a nuclear cytoplasmic ratio control of genome activation by which some repressive factors that are present in the cytoplasm, and that's been proposed to be histones, are titrated away by the replicating DNA, and that reduces this potential repressive factor, histones, and when you reach this concentration threshold, you get an activation, and for that you need cell division, replication of the DNA. And alternatively, there is a model of time where somehow, depending on the different species, there is a measurement of the of time by either regulation of the chromatin or translation of specific factors that reach a particular concentration that then allows the activation of the genome. So Henry decided to, instead of going on a genome-wide scale, try to use imaging approaches to look at the process of genome activation. And for that, we needed to label independent individual genes, and we use the Cas9 system where we use a dead Cas9 tagged with GFP and guided with a short guide RNA. And as a model, we use miR430 because we know it's one of the first zygotically transcribed genes, and it's actually a repetitive sequence of about 50 copies of this microRNA within 17 kb. We know that through collaboration with the Scarmeta lab, we know that this region is open, the level of the chromatin is strongly labeled by histone acetylation and by histone methylation. It's 3K4 trimethylation and K27 acetylation. And we know that this is not maternally deposited and is progressively activated early in development. So we decided to use this system to see whether we can visualize individual loci and then correlate it to the activation of the genome. And for that, you can just simply inject this cocktail into the embryo. And we can see this is now a single cell of a 3-hour embryo. And you can see 2 foci of the miR430 locus. And we know that that is miR430 because if we use a miR430 deletion control where we have knocked out 80 kb where the miR430 locus is, we don't see any signal. And in fact, now we can follow this foci in vivo. And this was actually, it's not a very high resolution, but it's the first movie that we got. So it has a sentimental value. So you can see through the process of development how we can follow the 2 individual loci and the dynamics of Cas9 binding. So you can see that now in this still where Cas9 is bound, it really, through the replication or mitosis, you can still see Cas9 associated with the chromosome. Then soon after nuclear envelope formation, you have ejection of Cas9, and we predict that this is through the replication of DNA where Cas9 gets evicted, and then 1 minute later, it gets bound again to the miR430 locus where it stays. And here, as you can see, the timing is quite fast, and the replication total time is about 20 minutes. So the first question we ask is quite simple, is do you have a global activation, simultaneous activation of many different genes, or do you have an individual activation that then gradually increase over time? So you have some genes initially activated versus a switch where you have a massive activation of the genome. And for that, we use two different approaches. We just label phosphopol2 that labels active RNA pol and prime RNA pol2, and we also label de novo transcription with labeled nucleotides using the click-it technology. And the problem is that you might be wondering, why don't you just use RNA-seq and just see which are the first genes activated? And the problem is that the majority of the genome is provided in the maternal form, and it really flats the de novo transcription. So we find that this is kind of a good method to look at de novo activation. So when we look over time for phosphopol2 as well as for the click-it that labels foci of incorporated nucleotides in the RNA, de novo transcription, you can see that the first foci that appear to be transcribed are actually those of the MIR430 locus that we can label with the Cas9, that then gradually get increased to then achieve global activation of the genome. And one of the interesting things that we didn't know about this click-it signal is actually every cell cycle, this click-it signal gets reset. So as soon as the nuclear envelope breaks down, all that signal gets diffused in the cytoplasm, and then you start again. So it really allows us to look at a chase of the click-it every cell cycle. So you can see that initially it's mainly the MIR430 loci activated, then you have some staining in the nucleus that is actually not background, because this is the background when we block zygotic transcription, and then you have a global burst of activation. So this is kind of what we think about the model. Second, we ask, well, can we also visualize, if we can look at individual foci, can we also visualize transcription in vivo? And for that, we use a molecular beacon, which is basically a hairpin that is quenched. It's a fluorescent hairpin that is quenched here. And the idea is upon transcription, so we can inject this beacon in the embryo, and upon transcription, this beacon can hybridize with the RNA, open, eliminate the quenching, and then fluoresce. And again, this is the first movie that we got. This is a 32-cell stage embryo where you can see now 2 cells labeled through the first cell division to the 64-cell stage. And what you will see in this movie is that you can then see the bursts of transcriptional activation in real time. So this movie is going back and forth. And now, through accumulating data across different time points, we can see when transcription first starts, whether it is really affected by the cell cycle, and what is the window of transcription during the cell cycle. So we have, and when I say we, Henry has really done a tremendous amount of work of quantifying across multiple movies and see how initial transcription of MIR430 begins really at the late stage of 64-cell stage, and it was thought that the activation really starts initially here. We can see initial stochastic activation in some cells, but not others. That increases in the time window as well as in the levels of accumulation of the signal. And by 512, you have a long period of activation of transcription. And as a control, we have used the MIR430 deletion mutant, and we don't see any increase in the signal of activation. So what we can see now from these results is that we already can predict that, well, the rapid cell cycle might be interfering with the activation, but it's not the main reason why you don't get activation of the genome, because we can see initial activation of MIR430 within the rapid cell cycle. So then what about the 2 other models, the nuclear cytoplasmic ratio and the timing model? And until now, we have really had hard times to block the cell cycle. So ideally, you would want to block the cell cycle and see whether you can get genome activation. However, if you use microtubule or cytoskeleton inhibitors, you can block cytokinesis, but you still get endoreplication. So that DNA really wants to replicate, so therefore it's being hard to really address that model. So the idea is that if we would block the cell cycle, we might be able to see early transcription if this is the true model. We should never see transcription if you really need to dilute those cytoplasmic repressors. And we would see transcription, but only later, even if we block the cell cycle, if the timing model is the correct one. So over the last year, actually, Philip Segerman has been studying the cell cycle in Xenopus, and they found that CHEK1 is required for the activation of the origin of replication, and it's actually able to block the cell cycle and block the replication of DNA in Xenopus. And when we use CHEK1 in the early embryo, you can see these are an uninjected and injected zebrafish embryo at the 4-cell stage. You can see how the uninjected embryo undergoes gastrulation, and there are many cells here, about 6,000 cells. While the CHEK1 stage stays in about the 4 to 6 cell stage when that is before the activation of the genome. So we decided to use this system to ask whether you get activation if you block the cell cycle, and whether the nuclear cytoplasmic ratio is the trigger or whether the timing is the trigger. So we use, this is the important panel, and this is the control. So what we can see is that the click-it signal is still dramatically upregulated when we block the cell cycle in these embryos, and compared to the control when we block zygotic transcription. And while it's lower than the activation that we see in the wild-type embryo, we think that blocking the cell cycle and blocking that changing in the nuclear cytoplasmic ratio still allows for the activation of the genome, suggesting that the embryo is measuring timing for this activation. So we think that that timer is basically either a translation of specific transcription factors or chromatin remodelers, or on the other hand, chromatin remodelers that are maternally provided that require some time in order to activate the DNA and modify the DNA. And we're currently testing those models, and we know that it's not entirely translation, because by 64 cells, the embryo has translated all the proteins that are needed for activation of the genome, but is not able to activate the full genome unless we give it extra time. So we are now inclined to believe a chromatin model, and we are testing different experiments by basically heterochronic transplant of the nucleus within development to understand when the genome becomes activated. If we take an old nucleus into an early embryo, whether that allows activation. And we actually have data that the chromatin plays a major role in this timing, because we have found an inhibitor of the reader that binds H3K27 acetylation called JQ1, that when we incubate the embryo with this drug, we fail to see global genome activation. So we think that part of the timing is one of the first signals that appear in the MIR430, which is K27 acetylation, that we can block with this inhibitor and block the activation. So the second part that we are really interested in is understand what are the mechanisms of post-transcriptional regulation within the embryo, and what is this post-transcriptional regulatory code. And just to illustrate, these are 3 maternally deposited mRNAs that are rapidly degraded upon zygotic genome activation, and we can actually dissect the different pathways that mediate this post-transcriptional regulation. Those that are dependent on transcription activation, because on the genome activation, because you see a block of mRNA decay if we block RNA pol2, while those that are maternally controlled, and those that are dependent on microRNAs, such as this that is dependent on MIR430, whose degradation is blocked when we block MIR430 activity, while other mRNAs are independent of microRNAs. So while microRNAs explain about 20% of the total post-transcriptional regulation of mRNAs, we still lack understanding of many of the mechanisms that mediate this post-transcriptional regulation at the level of decay, as well at the level of translation. So over the last years, we have been trying to decipher this regulatory code, And while we were very focused on the 3' UTR, recent experiments revealed that the coding sequence has an important role in the regulation of these transcripts, and that's what I'm going to tell you about. So to decipher this code, what we were using is a classical bashing approach, where you really want to test the regulatory activity across different mRNAs. You just take an mRNA, break it into pieces, and then test the regulatory activity of each piece. So instead of just doing one mRNA at a time, what we did was to look at the whole transcriptome. So just imagine that you take now all the mRNAs, break them into pieces, and then clone those pieces within a reporter mRNA. Depending on where you clone those pieces, you can look at regulatory activity within the 5' UTR, the coding sequence, or the 3' UTR. And by using a PCR assembly approach, we can basically clone a whole RNA-seq library anywhere that we want within a reporter mRNA. And because we are injecting RNA into the embryo, we can then in vitro transcribe that piece of linear DNA. And the complexity of these libraries is really only a few orders of magnitude lower than the Avogadro number. So it really gives us complexity to test every nucleotide within the genome. So I'll tell you now the story where we tested the regulatory sequence of coding sequences, and this is the work by Ariel Bassini, an extremely talented postdoc in the lab. So the idea is, well, if the ribosome is the most abundant reader of RNAs in the cell, could the coding sequence also, in addition of encode for proteins, convey regulatory signal in the cell? And there was original work from Jeff Kohler that shows that in yeast, actually the coding sequences can regulate mRNA half-life, and that was before we were actually, or during we were preparing this work. So the test that Ariel did, he basically had this library of codings or regions of the transcriptome cloned within a reporter that has an AUG and a stop codon, and then he asked, what is the differential stability of those mRNAs in the presence or in the absence of translation? And he blocked translation with a morpholino that binds to the AUG. And he did that in parallel in Xenopus and in zebrafish, with the idea of saying, well, if the ribosomes are really regulating mRNA stability, maybe we can see differential accumulation of codons. Some codons that are accumulated when you allow translation versus some codons that are degraded when you allow translation. So when he quantified the set of codons, and this is how we know the genetic code, he was really shocked to see that there are some codons that are accumulated, meaning that when those mRNAs are translated, those mRNAs are more stable if they have these codons than if they have the blue codons. So that was surprising and very exciting. And then, but alternatively, instead of codons, it could be just sequences that when you put them together, they really assemble a regulatory, for example, a microRNA target site. So is this really specific codons that are shaping mRNA stability, or is the sequence of the RNA independent of the codon? So he did a series of experiments, of which I'm showing you just one, where he basically takes the same sequence of an mRNA, except that with a single nucleotide insertion. So he compares two sequences that have the same sequence composition, except a single nucleotide insertion that changes the frame. And he designed those sequences in which, in one frame, it was mainly reading those blue destabilizing or potentially destabilizing codons, and in the other frame, without that nucleotide insertion, it was reading mainly those stabilizing codons or potentially stabilizing codons. So if it's the sequence, those two mRNAs should be equally regulated, equally stable and unstable. If it's the frame that the sequence is being read, those two mRNAs could have very different mRNA stability. And when he performed a northern blot after injecting those two mRNAs, he saw that those mRNAs where mainly stabilizing codons are read is much more stable than those mRNAs where mainly destabilizing codons are read. So that suggests that the coding sequence of an mRNA, when it's read by the ribosome, mediates differential mRNA stability. At the same time, in yeast, also Jeff Kohler showed that indeed differential mRNA half-life of mRNAs have a dependency of the coding sequence of those transcripts. So then this posed the question, well, could codon composition regulate the differential stability of these thousands of mRNAs in the embryo? So the idea is that we just measure how much mRNAs decay during the maternal to zygotic transition between 6 and 2 hours. And we just look at the fold change and do a cumulative distribution. So basically what you just need to know is that we are looking here at all the maternal mRNAs. This is an accumulation of all the mRNAs. And if we are able to select a group that is more stable or less stable, this curve should shift either here for more stable mRNAs or here for less stable mRNAs. So then we say if codons can really mediate differential mRNA stability, if we select mRNAs that have mainly these red codons and fewer blue codons, maybe those mRNAs are more stable. And if we select